Hello everyone, my name is Philip Nkwam. I am a final year student at the College of Medicine, University of Lagos, and I'm happy to be presenting our work on the reproducible and resource efficient resting state functional MRI analysis pipelines for dementia research in low resource setting. And this is a collaborative work. Now, in this paper, one of the things we solidified in the course of working on this project was extracted pretty much from this paper, Dementia in Africa. So we see how that racial differences begs more local research finding. What that means essentially is when we look at participants from high resource settings, maybe Caucasians, US, Canada, UK, and then we compare many of them with our local participants from low resource settings, or even more or less Africa to be precise. We sort of, in this paper, we sort of see a good amount of differences in phenotypical expressions and levels, t-tau and then p-tau levels, right, in African American also, and then Caucasian individuals. And so when it comes to normal cognition and then MCIs or AD, and so we see a good amount of disparity and differences when it comes to phenotypical expressions, right, physiological expressions from our local participants and Africans compared to Caucasians. And so it really begs that we are actually supposed to do a good amount of local research findings, right, in our own region to find out more insight about how on people and how on demographics, and then informs our approach to certain health solutions primarily. And one of the challenges we faced in the course of building solutions, particularly to our local demographics, had to do with, number one, we identified that for us to do imaging research, especially when it comes to PET or functional MRI or structural MRI, we realized that it takes a huge amount of computation. And those computations are not necessarily available for us here in low resource settings of Africa. And then another issue we identified had to do with the lack of trained personnel. And so we have very few amount of people actually doing neuroimaging analysis and building pipelines, sophisticated pipelines for our own region or our own demographics. And another thing we also identified had to do with poor infrastructure when it comes to the acquisition of our images. And that has to do with protocol optimization and other computational resource necessary. And one last thing also that we identified in the course of our work had to do with a need for standardization and reproducibility. And we haven't necessarily found any standardized workflow for image processing or pre-processing or post-processing, to be precise, for functional MRI, structural MRI, PETs, amyloid, or any of those. And so there is a huge absence of standardized workflows, or even at best, reproducible workflows in our region. But then in the course of this project, we decided to focus solely on functional MRI for a start, for our initial phase. And one of the things we looked at for our metrics extraction majorly has to do with the ALFF, the FELFF, and then the REO. The ALFF essentially is the amplitude of low frequency fluctuation. And a very simple way to explain what ALFF is when we look at different regions of the brain, there are signals that occur in a resting state, right? And so we want to see, while we are looking at signals occurring in a resting state, we want to see if we're going to notice any spike, right? Any anomaly in any brain region expressing anything more than the average signal we expect. Another thing is the fractional ELFF. It has to do with comparing the signal spike in one region to the other. So one signal spike from a region, voxel, to other signals, right? Other ELFF signals primarily. And then finally, we used the REO metrics. REO has to do with regional homogeneity. We want to see what regions of the brain have similar spikes, what regions of the brain are synced when it comes to their signals. Signals will mean time series signals from voxel regions of the brain. And as we all know, we register this in the anatomical plate, where we also identify atrophies in our data. And then we built a no-cost local high-performing computing infrastructure that was designed also as a part of a collaborative African neuroimaging training initiative. And this was cloud-based, so we had to do everything on the cloud. We do not depend on any local infrastructure or resource for this, and we had to make it reproducible. And so we want to ensure that anybody who picks up this script or this tool, right, can actually use it almost the same way we did. And then we leverage open source tools from the beginning to the end. We had to use all open source tools and then try to make it resource efficient. And so what that means essentially is our internet infrastructure is actually quite neat. And we try to make this run on a 10 megabyte per second internet bandwidth connection. And yeah, this program was spinned off this project. It's the Connecting program, had to do with the comprehensive neuroimaging analysis experience in resource constraint setting. So we're focused on enhancing research capacity for African neuroscientists, to be precise, and so we can actually contribute meaningfully to global scientific advancement. And then we leveraged on the data set, the Prevent AD data set, led by the team, Sylvia Valinov and Miguel. It's the pre-symptomatic evaluation of experimental novel treatment for AD. And so the data set came about. They tried to look at a good amount of participants with parents or relative that had Alzheimer's. Or so we essentially try to look at participants that are high risk of Alzheimer's disease and then follow them for a couple of years and gather data sets from them. But we utilized only 3 different time points for our preliminary phase. But the data set primarily has time points of up to 36 down to 48 months primarily. And then the tools, the open source tools we used were Neurodesk. So Neurodesk is a containerized modular platform that has a good amount of integration with Google Colab. And so we took Neurodesk and then integrated with Google Colab. So Google Colab leverages Jupyter Notebook and for also write our script, particularly for our analysis. And then we also used the FSL and AFNI-based models within Neurodesk environments to extract our functional connectivity metrics. And so this was the workflow. And so we take our images in bits format, the brain imaging data structure. We run quality control using MRI-QC, and then we pass them through our resource efficient pipeline. And the pre-processing steps primarily had to do with the conventional ones, slice timing correction for, essentially because we acquire images in slices, and then we want to ensure that all of the temporal, all of the slices are actually quite registered together, despite the differences in time points they were acquired. And so we want to put them in the same time point, and so we can have a good temporal resolution across board. And then we did motion correction, and motion correction many times we want to see in case there was any movement the patient actually did. We want to ensure that we rule that out. And so we sometimes, in our case, we registered with the 7th volume compared to the conventional ones of using it at the first or the average or the middle volume of the entire position. And then we did score stripping essentially to extract the brain region primarily, aside the skull and the brain matter primarily. And then we did spatial normalization and then band pass filtering, essentially to extract only signals between 0.01 to 0.08 hertz for analysis. And then we passed that through our post-processing where we extracted, for our preliminary phase, extracted our ALFF, functional ALFF, and then our RIO. This is the workflow for our entire pipeline. And then preliminary results had to do with only 17 subjects in the entire Prevent AD data set we had access to, about 75 data sets, subjects primarily. So we used 17 subjects for this particular preliminary phase to test run our tool. And the average pre-processing time, leveraging on to how to do with 20 minutes per subject per time point. Time point would mean from baseline to a follow-up of 12 months to a follow-up of 24 months. And this was our results. We see our ALFF map spread across our baseline, 12 months, and then 24 months with our signal levels down to our REHO metrics estimation. And I want to ensure that all of the documentation of our full analysis pipeline, want to ensure that you're put on protocols.io, ensuring transparency, and then put a bit of version control, and then facilitate reproducibility amongst LMIC researchers. And then a future direction, want to ensure that we do reliability analysis. So we're going to run a reproducible analysis across different teams, running the same pipeline to ensure we have consistency and generalizability of our platform. And then we also want to ensure that we leverage the entire prevented data set. We want to use this to on the full prevented data set, and also a good amount of Africa dementia study going on in our own region, because that's the goal. That's the primary goal, essentially for AD progression. It's a teamwork. We had supervisors and coordinators and participants from De Niro, Miguel, Hen, and then this project is very much a huge spin-off of the Connecting program, and then the MyLab Medical Artificial Intelligence Laboratory, and then Camera Africa. Thank you so much. A big thank you to my supervisor, Dr. Donana Zodo, Abdallah, Ethan, and every single person that made this work. Thank you very much.